I hope all of you have seen the link, YouTube link that I've shown you.
Yes,
I
ma'am.
posted a video in the group. It is regarding the DNA structure, composition of a DNA. So whoever has got any doubt, kindly clarify that. Hello? எல்லாருமே பார்த்துட்டீங்களா? Am I audible?
Ma'am, yes, ma'am, but I didn't, ma'am, I saw a YouTube video, ma'am, but on this one, 5 prime and 3 prime.
Yes, yes. Oh, you want me to repeat 5 prime and 3 prime?
No, I saw a YouTube video on 5 prime and 3 prime, ma'am. This one. And I got
Okay.
my doubts clear. Thank you.
Okay. See, this is what is your RNA molecule and your DNA molecule. So RNA is a single stranded structure and DNA is a double stranded structure, and you can see how the strands are twisted among each other. ரெண்டு strand முறுக்கி முறுக்கி இருக்கு. So that is what we call it as the helical. Helical means the two strands are getting twisted among themselves. This is called as the helix. And DNA is a double stranded structure, RNA is a single stranded structure. Now RNA or a DNA, if you see, they always start from a 5 prime and ends with a 3 prime. And in a DNA, if you see, one strand starts from a 5 prime, it goes like this and ends with a 3 prime. Whereas the other complementary strand, the opposite strand, will start from the 3 prime and it will end in 5 prime. So what does the prime here means? It is the 3 prime means it is the third carbon atom to which the hydroxyl group is attached that is getting exposed. 5 prime means it is the fifth carbon atom to which the phosphate group is attached is getting exposed. That is why, according to the functional group attached to that particular carbon that is getting exposed, we label the DNA ends as 5 prime or 3 prime. Now, when you look at the nitrogenous bases, in the center, as I told you the other day, the steps. நடுவுல ஒரு படிக்கட் மாதிரி இருக்கும். The படிக்கட்ல steps இருக்கும். So, in what is present in the step is, it is the nitrogenous bases. So, you have 4 type of nitrogen bases. It is classified as in 2 groups, major 2 groups, purines and pyrimidines. So, purines are guanine and adenine. Pyrimidines are cytosine, thymine, and uracil. So the thymine, which is present in the DNA, it is substituted by the uracil in the RNA molecule. So these are the purines and the pyrimidines. And why does the DNA forms a double helical structure? From where the 2 strands are coming? The concept of 2 strands are coming because it is so happening that the guanine molecule always form hydrogen bond with the cytosine. So guanine will be in one DNA, the cytosine will be in the other DNA. So the bonds will be established between the guanine and the cytosine. Cytosine-க்கும் guanine-க்கும் நடுவுல hydrogen bond நடக்கும். Because of this hydrogen bond, the guanine of one DNA strand, it is getting connected to the other DNA strand. So this is how the 2 polynucleotide strands are coming closer. In the end, polynucleotide strands, how they are coming? Polynucleotide strands means this one. I'm talking about the polymer. So all the nitrogenous bases are getting attached in a repeated manner and it forms a polymer. DNA is a polymer. So in the center, you can find the nitrogenous bases being present. In DNA, both the nitrogenous bases are attached with each other with the help of a hydrogen bond. Whereas in an RNA, if you see, all the nucleotides are free. எல்லா நியூக்ளியோடைடும் RNA ஃப்ரீயா இருக்கும். ஏன்னா RNA complementary strand கிடையாது. In an RNA, you don't have the complementary strand. In DNA only you have 2 strands. So one strand nucleotide will form the hydrogen bond with the opposite strands nucleotide. So cytosine always binds with guanine, adenine always binds with thymine. And the bond number between the adenine and the thymine is, adenine and thymine will have 2 hydrogen bond, cytosine and guanine will be able to have 3 hydrogen bond. Now, why should here 3 hydrogen bonds being present? What decides the formation of the bond? It's a question to you all. Anyone can answer. What decides the formation of the bond? Why cytosine and guanine are only able to form the hydrogen bond among themselves?
Ma'am, because they have three bond.
Three bond. I'm asking you why the three bond is being formed. Okay, let's come back to your chemistry. Why do you think A and B are interacting? Why should A and B interact to give you C? Why a sodium and a chlorine should interact to form sodium chloride? Why
To give
hydrogen?
stability, ma'am.
Stability of what?
Stability of
Electric
the molecules.
stability of the particular atom. By
Yes,
bonding
ma'am.
with another atom, it can get the
Yes,
stability.
ma'am.
Correct.
Yes,
May I
ma'am.
know your name?
Sukesh, ma'am.
Thank you, Sukesh. See, listen here. A is interacting with the B to form C. Why A and B should interact? Because A is having some disturbance in its energy level. A may have a lesser energy. Why B is getting disturbed? Because B is having a higher energy. So A and B will come closer, and the A needs energy. B does not want energy. It wants to give it out. So now A and B, what it will do? Both of them will come to an understanding. Here, both of them means I'm talking about the atom. So one atom is having a less energy, another atom is having a high energy. So now, in case of an ionic bond, from the high level energy is completely getting transferred to the low level energy group. In ionic bond, in covalent bond, what is happening? The energy is being shared between the two atoms. So you get a product called a C, which is stable. See, any chemical in the whole world, they undergo a chemical reaction to satisfy their energy level at its atomic state. The atom must be stable. What denotes the stability of the atom? The proton, the nucleus should contain proton and this one neutrons. The proton number must be balanced with the electron number. Now electrons will be rotating the nucleus. So this is the construction of an atom. Now in this construct, if a proton is not there, an electron is not there, or a neutron is not there, such an atom, it is unstable. So to satisfy its instability, the molecule definitely it will go and have the energy level with the other atom. Now for this only the chemical reactions are being formed. So why does the cytosine and guanine form 3 hydrogen bonds? Because there are 3 functional groups, and because there is an involvement of only 3 functional groups among this. So what will be the 3 functional? You see cytosine. Cytosine is having an oxygen. Oxygen is a highly electronegative molecule. Nitrogen. Nitrogen is also an electronegative molecule. So here one nitrogen is satisfied. Nitrogen valency is what? 3. So the 2 hydrogen here, and it is forming one more bond with a carbon. Whereas these 2 nitrogen, they require an electron, sorry, they require a proton. And this oxygen also requires a proton. So that is why the cytosine is able to form a bond with guanine. Now, why not with adenine? Because in adenine, the functional groups are completely different. So cytosine will not form a long-term bond with adenine. See, adenine is one such purine that it can bind with thymine and also cytosine. Predominantly, adenine is binding with thymine, but it cannot bind with 100% for a long time duration. It cannot form with cytosine. So, cytosine and guanine bonding only is a stable bonding. Any atom to satisfy the energy level, they can form the bond with any other atom. But stability for a long term is a big question mark. So when cytosine binds with the guanine, you will have a more stable interaction between them. When adenine and thymine are forming two hydrogen bond, because in thymine, if you see, there are two oxygen molecules are there, so that is why adenine and thymine are able to form the bonds, that is two hydrogen bonds. So please remember, hydrogen bonds are formed to satisfy the energy level of both cytosine and guanine. So whichever purine and whichever pyrimidine are able to satisfy the energy level, they will form the complementary hydrogen bonds and they form the double helical structure. Now coming to the backbone of the DNA, the rim of the DNA, it is made up of phosphate and sugar molecule. Now, the sugar interacts with the nitrogenous base to form a structure called as nucleosides. And when the sugar molecule to the fifth carbon atom, when the phosphate group is attached, then it is called as nucleotides. So what is nucleotide? Nitrogenous base plus the sugar is a nucleoside. What is a nucleotide? Nitrogenous base plus sugar plus the phosphate group. When all the three things are present together, it is called as nucleotide. Simple, you can describe nucleotide as a nucleoside plus a phosphate group is called as a nucleotide. And this is how the nucleotides are formed, and the nucleotides are only going to be added onto your DNA. So along with the nitrogen base, sugar and the phosphate comes inside. So the phosphate of one group, the new coming nucleotide, will form the diphospho bond with the existing sugar's hydroxyl group. டிஎன்ஏல ஏற்கனவே இருக்கக்கூடிய சுகர் மாலிக்யூலோட ஹைட்ராக்சில் குரூப் கூட தான் இன்கம்மிங் phosphate group will form the bond. I'll repeat it. What is nucleoside? Nucleoside is nothing but the nitrogenous base plus the sugar molecule. Now this is nucleoside. What is nucleotide? Nucleotide is nothing but the nitrogenous bases plus the sugar, which is a nucleoside, and nucleoside plus phosphate is the nucleotide. Now you would have studied something called as DNA replication. See, this is a DNA replication which I will be talking to you now. So when the DNA is growing, when the new DNA are getting synthesized over here, the to already existing nucleotide, the incoming nucleotide will be bound. So that is what I'm telling you here. This the incoming phosphate group will bind with the existing hydroxyl group, and the enzyme involved in linking one nucleotide with another nucleotide with the help of a phosphodiester bond, what is that enzyme called as?
Ma'am, can you repeat the question? I didn't hear you quite clearly.
The question is, name the enzyme which establishes the phosphodiester bond between the existing sugar and the incoming phosphate. What is the enzyme called as?
Ma'am, ma'am, DNA polymerase?
Yeah, it is DNA polymerase, as simple as that.
I thought
And
this is not nucleobases.
what, Revant?
No, ma'am, no, I got nothing, ma'am, nothing, ma'am. Sorry, ma'am, nothing.
That's why I was giving you clue. The DNA, when the nucleotides are attached with each other, they're called as polymers. So what is the enzyme involved here? DNA polymerase. So
DNA
DNA
polymerase.
polymerase will establish the bond between the existing hydroxyl group and the incoming phosphate group. So it will form the bond. So when the bonds are being formed, the DNA will polymer. The DNA will undergo the replication state. Okay, now how does such DNA are present inside our cell is that is, you have a set of protein. So these are the green color ball like structures of the protein. Over the protein, the DNA is wound. Wound means DNA will be rotated over the protein. So protein center or pillar மாதிரி நினைச்சுக்கோங்களேன். பில்லர் மேல எப்படி உங்களுக்கு ஒரு plant grows on, the creeper grows on, same way here. Just imagine the post is a protein, the pillar is a protein. Over the protein, the DNA is rotating. The DNA is attached. So this is the nature of the DNA. This is how the DNA is present inside the cell. So see, in our body, when you remove the DNA from all the cells in our body system, we can go from earth to moon 2 times. So much amount of DNA we have in when present inside all our cell. But when a cell, if you take, the cell itself is a small size. Inside that cell, the nucleus is present, and inside the nucleus, you can see here, inside the nucleus only you have got the DNA is present. So how does the DNA is present in such a small space? It is found in a folded manner. Just imagine, in your dress cupboard, you are not folding the clothes, you're just throwing the clothes inside the shelf and you're closing the shelf. For you, the shelf is being filled. But will you be able to accommodate more number of clothes? Definitely the answer is no. So that is why you fold your clothes and you keep it in the cupboard. So whenever you want that particular dress, when you're folding it and neatly keeping it arranged, you can pull out that particular dress, you can wear it. There is a time consumption is there. Just like that, when the DNA has to express its protein, that time the DNA must be picked out by certain enzymes. Now, the DNA is going to be folded. It is easy for that enzyme to come and locate that particular DNA, and it can start the protein expression. So that is why the DNA is present inside the small cell in order to occupy the small cell, and in order for the other enzymes to come and bind to the DNA is folded. So this is the first level of folding. This is called as the 30 nanometers fiber, and then you go on to the increase of the folding. So you can find when all these DNA, they form like a bundle. Now this bundle is what we call it as a chromosome. The relaxed form of a eukaryotic DNA, when it is interacting with the protein, it is called as chromatin reticulum. The
Chromatin.
same chromatin reticulum, when they are highly condensed, when they are interacting with so many proteins, then it will form this X-shaped chromosome. And this is what is the actual structure of your chromosomes. And this is what we call it as a 30 nanometers fiber. So the folding keeps on increasing. The DNA is tightly packed inside the chromosome. Now, when the DNA, when the cell is there in the, uh, when the DNA is there in the G1 phase, the DNA is relaxed. In the S phase, DNA is relaxed. They will be present as chromatin reticulum. So G1, S and G2 phase, in all the three phases, DNA will be present in the chromatin reticulum state, relaxed state. When it comes to the mitosis, the entire DNA present inside the cell must undergo a condensation step to form chromosomes. So chromosomes are the condensed nature of the existing DNA. And when the DNA interacts with the protein alone, it is called as chromatin reticulum. Clear? Any doubts?
No, ma'am.
Ma'am, can you explain the chromatin reticulum part again, ma'am?
Chromatin reticulum is a relaxed state. This
Okay, ma'am.
state of DNA, when it is interacting with certain protein called as histone protein, this state is called as a relaxed state. Now look at here. It is tightly folded.
Yes,
Many
ma'am.
DNA are brought closer. Now, this stage we call it as the chromosome. The condensed state of the chromatin reticulum is called as the chromosome. The relaxed state of the DNA is called as your uh, chromatin reticulum. So,
Okay,
what
ma'am.
is reticulum? Framework of segments. So, here the DNA is present loosely packed with the protein. This is chromatin reticulum, and on staining, chromatin reticulum takes up more stain. It looks bright in color. That's why it's called as chromatin. Chromatin is a Greek term, means chromos means color. So the most colorful part of your cell.
Yes, ma'am.
And this is how the chromosomes are attached. Chromosomes will have a centromere, a central ball-like structure over which two sister chromatid arms will be arranged. சென்டர்ல சென்ட்ரோமியர் இருக்கும், ரெண்டு சென்ட்ரோமியர் மேல கீழேயும் வந்து chromosomes arms will be attached. So when the cell is in the G1 phase, S phase and in the G2 phase, the cell, the DNA is present as chromatin reticulum. Only during the M phase, the DNA is getting condensed. The chromatin reticulum is getting condensed into a structure called as the chromosomes. So when DNA is present as a chromosome, segregation between the cells is easy.
Ma'am,
Uh,
ma'am, I have a doubt. Like
yes.
about the centromere, there is another like term, kinetochore. Ma'am, during the like
What?
separation,
Vikram, your voice is breaking.
ma'am, I
Yes.
ma'am, ma'am, there is another term called kinetochore.
Yeah.
Ma'am, during the separation, the
Yeah, go ahead.
ma'am, during the separation, the spindle fibers should like where will we attach? A kinetochore or a centromere?
Both.
Both.
Yes. See, dear children, this is a little bit advanced. Maybe in cell biology, a sir would be discussing about this. See, Vikram, the kinetochores will be diffusely associated with the chromosome. They
Okay.
will not, when you see the, see, let me get the picture of it. Can you see here?
Yes, ma'am.
See, this entire region is called a centromere region. In this region, you've got another protein complex, and that protein complex is called as kinetochore. So, the kinetochore only the spindle fibers, the aster rays or the microtubules will come and attach. It is a point for the attachment of the microtubules.
So, ma'am, so in general, like in speaking, so like the spindle fiber will attach at the centromere.
Uh,
Okay.
and you see here,
Yes, ma'am.
this is not clear. See here. So these are your spindle fibers. They are attached to the chromosome.
Yes, ma'am.
Okay. Now the center arranged chromosomes have to move to the periphery, to the
Yes, ma'am.
cell end. So here you will be having the centrioles. The centrioles will rotate. When the
Okay.
centrioles are rotating, it is actually going to chop your microtubules. One end of the microtubule is attached where? To the kinetochore. This you can see here, kinetochore.
Yes, ma'am.
So the rope-like structure that is attached to one end to centriole and another end to your to the kinetochore, due to the breakage of the microtubule, the length is going to shorten. So what
Okay.
automatically it will do? It will pull the chromosome near to the centrioles.
Okay, ma'am.
So for the movement of the chromosomes or the distribution of the chromosome from the center to the periphery, you require the spindle fiber, and the place where the spindle fiber attaches itself to the chromosome is called as kinetochore.
Okay, ma'am.
Ma'am, one doubt.
Yes, Revant.
To confirm, ma'am, like all the chromosomes will be to get the chromosome to the peripherals. The spindle fibers will actually like pull them to the peripherals, ma'am, and then the point where the spindle fibers are attached to the chromosome is called kinetochrome.
Yes.
Okay, ma'am, got it. Thank you.
I'll see some good video and I'll post it for you in your group. You can see beautifully how the chromosomes will move to the periphery. I'll get it. Now we go on to our other thing. Okay. Now what is the main function of the DNA?
The main function of a DNA is to store information, ma'am, about the body.
What information?
Information about this one like the antibodies of like how to fight cold or fever. Like if the cell if the cell divides, now the cell also has the same knowledge of the previous cell, which knew how to fight any fever or cold. It stores information.
Okay. So a
And
DNA
also,
contains a sequence of nucleotide which participate in a protein synthesis. One. The second thing is the DNA expresses the protein and that gives the phenotypic character of a person. Phenotypic character means what? The character what you can observe visibly. Now here I've taken you the example of the eye color. So you can find different eye colors, but all these belongs to the color of the eye. So color of the eye is the gene. The alternative form of the gene is called as an allele. And what is a gene? Can you tell me what is a gene?
Sequence of DNA, ma'am.
Sequence of DNA is called as a gene.
No, ma'am. It's like functional unit.
So what will be the terminology?
ma'am, it's this one, ma'am.
Genome.
Gene actually contains a hereditary sequence, like from the
The
parents.
question is what is a gene?
This one. Ma'am, it's a sequence. It's a nuclear. It's a sequence of
Unit
nucleotides.
of inheritance.
Ma'am, sequence of nucleotides.
Sequence of nucleotide. A sequence of a nucleotide what? Complete.
Uh,
Of a chromosome.
What am I typing here?
Sequence.
Sequence of a nucleotide of a DNA which expresses a protein is called as a gene.
Oh yes, ma'am.
See, when
Oh
you
yes.
just have a sequence like ATGC, that is a DNA strand.
Yes, ma'am.
But what is a gene? A gene is a sequence of the nucleotide which participate in a protein synthesis. It is able to express the protein. If AGTC is able to express a protein, then that sequence becomes a gene. Now, coming to this eye color, you have a gene to express the protein, and the protein gives a color to the eye. Right? Now, this is a
Yes, ma'am.
gene. Now, in all the four eyes, if you see, there are a variety of color are there. So, the person is having a eye color coding gene is present. Now, what is an allele? If you see an eye color, there is a brown color, blue color, emerald, gray. So, there is a difference in the color of the eye. There is a difference in the color of the eye color pigment. Now, what is an allele is? What have I written here?
Sequence of a nucleotide of a DNA which expresses a protein with alternative protein expressions. Oh,
That means, see, listen here, that means they're all eye color coding gene, but they have a slight change in the nucleotide sequence. The nucleotide sequence, there might be a change with one or two nucleotides. So the protein that they are going to express, which is going to give the color of the eye, there the difference happens. That is why in your textbook they have put as allele is an alternative form of a gene. Alternative form. It is eye color coding gene. In the eye color coding gene DNA sequence, there is a change. What is a dominant eye color for human being?
black.
Ah,
Ma'am, there is actually one fact. I don't know if it's true or not, ma'am. But
ah.
every person with the eye color of blue actually originated from a single person's ma'am. Single person eye color.
Yes, that is why we tell dominant color.
Yes, ma'am.
See, here the sequence must be ATTTGCT. If
Yes,
this is a
ma'am.
sequence, is there eye color is black. Dominant eye color of every human being is black. But in some person there is a brown color. When you analyze the sequence, you can find that there is a change here. See, in T and G, there is a substitution. Only there is a substitution in the T and G, not anywhere else. So the second sequence also produces eye color, but the color is brown and not black. The dominant eye color, what will you say dominant? The number of gene that is present in a population in majority number. ஒரு இந்தியா எடுத்துக்கிட்டீங்கன்னா throughout India, when you do the analysis, they'll have one character. So for Indian population, that is the dominant gene. For the other people, when they have a slight variation, that variation occurs where? The variation occurs in the sequence of the DNA because the protein is synthesized from the DNA. So if the DNA sequence, even though there is a slight change, the protein produces eye color. But what is the color? It is a different color. It is a brown color. So what is an allele is? An allele is an DNA sequence with an alternative or a slight altered DNA nucleotide sequence which expresses an alternative protein is called as an allele. Gene in a sequence of a nucleotide that is going to express a protein is called as a gene. In that gene, when you have variety of gene, so how are you saying 6 variety belongs to one gene? When you do the analysis of the sequence, you can find like this is the dominant eye color gene and this is the uh, allele of that gene. So eye color gene means the sequence is ATTTGCT. இது இந்த சீக்வன்ஸ் இருந்தா ஐ கலர் பிளாக். ஆனா பிரவுன் கலர் பர்சனுக்கு அங்க என்ன சேஞ்ச் இருக்கும்? there will be one or two nucleotide changes there. It is the same protein being present with some slight alteration. So sequence-லயும் alterations இருக்கும். The same alterations can be exhibited in the proteins also. Then such things, such DNA sequences are called as an allele. Allele புரியுதா? Allele and gene என்னன்னு?
Yes, ma'am.
Okay.
Ma'am,
Ma'am,
in
I
India,
have
Yes.
in India, ma'am, the dominant eye color is black, ma'am.
Yes, it is
Yes,
black.
but in Finland and all, ma'am, the dominant eye color is blue. Like there,
Yeah.
if you have a black color eye, you possess allele gene.
For Finland, that is a mutant.
Yes, ma'am.
oh, okay, ma'am. So, ma'am, as
Ah.
we know that like the blue color is very rare in nature. Okay? So can we say that the like the blue eyes, the people who are having blue eyes, uh, like they are mutant? Like it is.
See, see, mutant is a terminology which you have to use when there is an alternative body character totally.
Okay.
But as you say, at one point we can tell all these eye colors are mutants only.
Oh,
Mutant is what? Mutation is what? Sudden change in the DNA sequence and the altered DNA has to express an altered protein. That is what is mutation.
yes, ma'am.
So dominant eye color is black. Brown, blue, emerald, grey, they are the mutant form of that particular color, that particular dominant gene.
Okay, ma'am.
Okay, so
Yes,
I
ma'am.
hope this is clear to all of you. Now, how many chromosomes are there in human beings?
23.
23 pairs are there.
Yeah.
Now, they have taken the photos of the individual chromosome and you arrange the chromosomes because we have got 23 pair. Every chromosome will have its own pair. So this technique is called as karyotyping. When you understand the number of the chromosomes present inside a cell, it is called as karyotyping. So karyotyping will give you a correct definition about the total gene present or associated with the cell. And this is called as genome. What is a genome? The genome is the total number of gene present inside the cell. Now where the gene is present, it is present associated with the chromosome. So, when you want to identify a defective gene in a patient, or when you want to see what is the dominant gene in the person, first you remove all the 23 pairs of chromosomes from the cell and you arrange them. How will you arrange them? By taking the size of the chromosome. Now, how the size of the chromosomes are determined based on the number of genes that they have, or what is the total length of DNA present in that particular chromosome. According to the length of the DNA, the chromosomes are classified into 23 pairs of chromosomes. So you isolate a chromosome from the cell, from a cell, and you arrange it and you view it microscopically. Then the technique is called as karyotyping. So when you do the karyotyping, you will know in which particular chromosome what gene is going to be present. See, and this is very, very useful for you to identify genetic disorders. Now, see here, one means this is the size of the first chromosome. Suppose there is a deletion happening. Deletion means what? A particular DNA sequence is lost. So when the particular DNA sequence is lost, the size of the first chromosome will decrease. It will not increase. It will decrease. And you know this is the first chromosome. So what interpretation you can give? There is a gene deletion. There is a loss of nucleotide sequences that has reduced the size of the chromosome. Suppose there is an addition, a duplication is happening. Then that means what? The particular DNA sequence is present in the chromosome in extra number. In high level, it is present. That is why the length of the second chromosome increases. So either decrease in the chromosome size or increase in the chromosome size, you would have studied as mutation. And mutation causes genetic disorders. So when you identify the chromosomes, we can very easily tell whether all the genes are intact or there is a deletion or an addition of genes in the chromosome. This is what we call it as karyotyping. So karyotyping is a technique where you remove the chromosomes from the cell and arrange it on the slide and you pair the chromosomes. Why should you pair? Because in our cell we have got 23 pairs. So 23 pairs of chromosomes will be arranged like this, and this technique is called as karyotyping. Is this clear?
Yes, ma'am. How,
Yes,
ma'am?
ma'am.
Yes. Who else
Revati,
has got a
ma'am.
doubt?
In this karyotyping, ma'am, like how will you know like this is the first chromosome, this is the second chromosome? Ma'am, is it arranged via the height of the chromosomes, ma'am? Like I have noticed that the tallest is 1 and the shortest is the 23rd. Is it arranged by the height, ma'am, or is there any separate technique?
Now, 1 second. Mahalakshmi, ma'am. Students are totally excited. It is disturbing me in my class. Please try to understand that you are the not only class who's sitting over here. I am taking the class for 40 years. You can all have fun. Please be softer. Nothing regarding another. நிஜமாவே class concentrate பண்ண முடியல. எல்லாமே பண்ண வாடி சொல்லவா பண்ணுங்க. I'm sorry, I'll just show you one small picture. Now, can you see this?
Yes, ma'am.
Now it is clear, no? See, every chromosome, you have a dark band and a light band. So, for a first chromosome, how many dark band and how many light bands are there? They'll count. What is the band? Band is the place where the nucleotides are tightly coiled. So, the number of nucleotide present in this particular segment is more. Why this area is light? Because the number of nucleotide present in the area is less. So, we can tell the lightly colored area as interband and the darkly colored area is a band. So, in a karyotyping, they'll identify the chromosomes based on counting the bands. See, this is how when you isolate the chromosome from the cell, this is how your chromosome will look like. Now, as a technician, we have to match the chromosome. So, how will you match the chromosome? For every chromosome, you will count the number of bands, which are all the darkly colored area that they will identify. Based on the colored area, they will tell what is this particular type of chromosome. Now, you keep on pairing the chromosome based on the number of the band. Now, for suppose, for example, 14th, you have a 14th chromosome. It is must be like this. Now, you are counting it, you are getting an extra band. That means what? Some other DNA has got attached here, or the 14th chromosome have got multiplied. That particular gene has increased its number. So, the 14th chromosome length will be bigger. Now there is another chromosome. The first chromosome is like this. See, this is the first chromosome. It's a very elongated chromosome. Now in the first chromosome, you are counting the number of band. So there is a standard. You have told, okay, this is my first chromosome. You have kept. The second chromosome also, of the pair of the chromosome, you are counting the band. It is not matching, and you find you have matched all the chromosomes. 22 chromosomes you have finished matching. Now the first chromosome, one chromosome you have identified. The second chromosome is there in this mixture. How will you do? Why you are not able to identify? Because from the standard chromosome, the band number is varying. Why there is a variation? The gene would have got duplication or gene would have got deleted. So it is the first chromosome. This is how they identify the chromosomes, depending upon the band. What is a band? Band is a place where you have the genes. Genes எங்க இருக்குமோ அது பேர் தான் bandன்னு சொல்லுவாங்க. ரெண்டு பேண்டுக்கு நடுவுல இருக்கிற செக்மெண்ட் பேரு interband. Chromosomes identification is done based on the band. இப்போ second chromosome பாத்தீங்கன்னா you can count 1, 2, 3, 4, 5, 6, 7. 8, 9, 10, 11, 12, 13, 14. So 14 bands இங்க இருக்கு. For whichever chromosome you have 14 bands, you pair them together. You will put it under the second chromosome category. Now this is clear?
Yes, ma'am.
Ma'am, I have one more question also. I'm like, if there are more than like 23 like chromosomes, then only we will say people is autistic or disabled.
That is operation. See, operations will be two type. One is the number difference will be there. Another operation is within the chromosome itself.
Okay.
The chromosome bands would have got deleted. That is also an operation. You have studied as euploidy and aneuploidy. Ploidies.
Yes, ma'am. Yes, ma'am.
Chromosome disturbances, operations.
Yes, ma'am.
See, all these things, what I'm telling you is now you will study this in detailed in genetics. Okay, I just wanted to give an introduction with the DNA, what you have, what can you do? That is what I'm trying to explain it to you now. So, function of a DNA, it is going to determine, as you all said, it is an hereditary material. The DNA from the parent cell, it is getting transferred to the daughter or the son cell. This is what is called as heredity. So one function of a DNA is to get transferred from one generation to another generation. And the second function of a DNA is it is controlling the cellular functions. The third thing is the expected protein is secreted by the DNA. So these are the three functions of the DNA. First is hereditary. Second is it determines controls the function of the cell. The third thing is it is able to produce the protein what the cell needs. Okay, now let's talk a little bit about the just 10 minutes, I'll finish it. Okay, so now I'm going to talk to you about the protein synthesis. See, so during the protein synthesis, the steps involved are the DNA which is going to participate in the protein. First it will undergo DNA replication, it will increase the number. After the DNA has got replicated, the DNA is getting converted into an RNA. RNA is getting converted into a protein. See, the nucleotides between the DNA and the RNA will be identical, except uracil and thymine. All the other nucleotides will be the same. For this, for you to understand or remember, let me give you an example. You all know we've got variety of Tamil. Tamil is a single Tamil language, but when you go to Coimbatore, the people who speak in Coimbatore Tamil will be different. In Chennai, people use a different accent to speak a Tamil language. When you go to Madurai, in Madurai, the way in which people are talking Tamil, the using Tamil will be different. So here, the change is between the language itself. It is not language to language, you're not changing. The change is between the language. So here, what is present as a DNA is now getting converted into an RNA. All the deoxyribose sugar are replaced by the ribose sugar, and wherever thymine is present, that will be substituted by the uracil. So a nucleotide is getting converted into another form of nucleotide. That is why you have called this mechanism or the step as transcription process. Now you take an RNA to a protein. RNA is made up of nucleotide. Protein is made up of amino acid. So the chemical composition of the RNA and the protein are different. I hope you all accept with me.
Yes,
So
ma'am.
when this is example is you are having a statement in English, you are translating it to Tamil. So language to language translation. Here, RM, which are all as a nucleotides that are getting replaced by the amino acids. That is why this mechanism or the process is called as translation. And this entire process, DNA replication, yielding to the RNA synthesis, from the RNA when the proteins are being produced, the entire concept is called as central dogma. Central dogma is a French term, means principle of life. So what is the principle? How are we surviving? How are we thinking? How are we moving our hand? So all these things are the function generated by us as a eukaryotic system. For that function, you need the cellular DNA. So the DNA will replicate first. The replicated DNA will undergo the transcription to produce the variety of RNA molecule. Now, all the RNA together, they will undergo the translation process and they start producing the protein. This is what is the central dogma. Now, picture if you see in the chromosome or the chromatin reticulum, the DNA is present. To the DNA, RNA replication will happen, and the RNA polymerase comes and bind. RNA synthesize the mRNA molecule. mRNA comes out. See, this is the entire thing is the nucleus, and within the nucleus you have some gaps. These are called as nuclear pore. So through the nuclear pore, the mRNA comes out. The mRNA is captured by the endoplasmic ribosomes, which are attached, in turn attached to the endoplasmic reticulum, and you have the poly amino acids being linked, and amino acids are linked together with the help of a bond called as the peptide bond. Now, each and every amino acid, each and every ball here represents an amino acid. All the amino acids are attached with each other and they form the polypeptide chain. Many such polypeptide chain interact with each other to produce the functional protein. See, this is a small video of DNA, which again you will study all these things in detail. So this is how the DNA unwinds and the daughter DNA strands are synthesized. So like this, from one DNA strand, you can get two copies of the same DNA. This is the concept is called as DNA replication. Okay. Now, what happens to the protein? The protein that is getting synthesized in the cytoplasm is then getting packed into the Golgi complex. I hope you all know what is a Golgi complex. It's a subcellular organelle inside our cell.
Yes, ma'am.
Now, this Golgi complex carrying the protein will come and fuse with our cell membrane, and the proteins are secreted outside. So, this is the mechanism. I'll repeat once again. The synthesized protein will enter into a vesicle, a bag-like structure. Inside the bag, the protein is going to be present. Now, this protein will form the Golgi complex. From the Golgi complex, a vesicle will detach. That vesicle fuses with the cell membrane and it will release all its content outside. So this is how content here means it is the protein that is present inside the vehicle. So this protein, when it is getting secreted outside the cell, it is called as extracellular protein. So this is how it happens. So from the endoplasmic reticulum, the proteins will get transferred to the Golgi complex. From the Golgi complex, the protein is sent outside. This is clear?
Yes, ma'am.
Whatever protein is secreted outside the cell, according to that, the other cells will behave. On the whole, an organ will start functioning. Organ will start exhibiting its function. Is it clear?
Yes, ma'am.
Okay. See, I'll
Ma'am, so
give
like whenever cells getting destroyed, like now this kind of possibly happens to like alert other cells that there is a germ cell in between. So this is the one of the type, ma'am, that cells communicate with each other.
cells communicate with each other with these signals. That and all
Yes,
I'll
ma'am.
talk to you later on. Now it is too
Okay.
early for you. So
Okay.
whatever protein the cell is going to secrete, yes, the cell communicates with each other. Or the protein will start exhibiting a function inside the cell. Is it clear?
Yes, ma'am.
See, today's class, what I wanted to tell you from yesterday to today is that the cell take up the nutrient undergoes metabolism. Due to the metabolism, energy is produced. With the energy, the cell is able to multiply and synthesize the protein. In today's class, yesterday's class in the end, we saw about the DNA structure. So it is the DNA that is replicating, it is getting converted into the RNA molecule and the process called as transcription. From the RNA, the proteins are produced and that is called as translation. So
Yes, ma'am.
this is how the cell is going to produce the protein of our interest. Is this clear to everyone?
Yes, ma'am.
Yes, ma'am.
Okay, ma'am.
Yes, ma'am.
Thank you all. You can leave today. Tomorrow you don't have any class. Okay, now you'll all meet on Monday only. Monday only we'll all meet. On Monday, I'm going to talk to you about biotechnology, the steps involved in biotechnology. And I will be creating an email ID for your class in where all the materials will be, that is videos will be posted. And I've already posted the PPT in your Google Classroom, so you can go through that. Is it clear?
Yes, ma'am.
Yes,
Yes,
ma'am.
Okay,
ma'am.
ma'am. Thank you.
Yes,
See,
ma'am.
dear students, again I'm telling you, anyone has got any doubt, text me or you can post it in your group also, WhatsApp group also. I will be able to give you the answer.
Okay.
but please be thorough with these basics. And all these transcription, translation, DNA replication, everything you can study in detail in your cell biology class. Thank
Yes,
you.
ma'am.
Okay, ma'am.
Thank you, ma'am.
Thank you.
Ma'am,
Yes.
ma'am, I have doubt.